Welcome, medical students, to today's lecture on the fascinating topic of aspartate and its role in biochemistry and medicine. Aspartate, also known as L-aspartic acid, is one of the 20 amino acids that serve as the building blocks of proteins in living organisms. It plays crucial roles in various metabolic pathways and has clinical significance in certain diseases. In this lecture, we will delve into the intricacies of aspartate metabolism and explore its diverse functions in the body. So let's begin our journey into the world of aspartate. Aspartate transaminase, or AST, is an enzyme that catalyzes the reversible transfer of the amino group from aspartate to alpha-ketoglutarate, yielding oxaloacetate and glutamate. This reaction, known as transamination, is vital for the interconversion of amino acids and the synthesis of non-essential amino acids. AST is found in various tissues, with elevated levels in the liver, heart, and skeletal muscles. Measurement of circulating AST levels is commonly used as a diagnostic marker for liver and heart diseases. Deficiency of aspartate aminotransferase, also referred to as AST deficiency, is a rare genetic disorder characterized by the reduced or absent activity of AST in the body. This condition can manifest with developmental delays, intellectual disabilities, and various neurological abnormalities. The absence of AST leads to disruptions in the metabolism of aspartate and other amino acids, resulting in imbalances in neurotransmitter levels and impaired brain function. Aspartate racemase is an enzyme that catalyzes the racemization of aspartate, converting L-aspartate into D-aspartate. This process is important for the biosynthesis of various D-amino acids, which have been recognized as regulatory molecules in the central nervous system. D-aspartate, in particular, has been implicated in neural development, neurotransmission, and synaptic plasticity. Fumarate biosynthesis from aspartate is a critical step in the tricarboxylic acid, TCA, cycle, also known as the Krebs cycle or citric acid cycle. In this metabolic pathway, aspartate is transformed into fumarate through a series of reactions involving the action of enzymes such as aspartate aminotransferase, cytosolic aspartate aminotransferase, and fumarate hydratase. This conversion generates energy-rich molecules in the form of ATP and NAD and plays a crucial role in cellular respiration. Aspartic acid decarboxylase, or DDC, is an enzyme that catalyzes the decarboxylation of L-aspartic acid, forming beta-alanine. This reaction is an important step in the biosynthesis of certain neurotransmitters, such as gamma-aminobutyric acid, GABA, and beta-alanine itself. DDC activity is particularly abundant in neurons and is implicated in the regulation of neurotransmitter levels, neuronal excitability, and neurodegenerative diseases. Aspartate and arginine are amino acids that often interact in various biological processes. The aspartate-arginine interaction plays a crucial role in protein-protein interactions, DNA binding, and enzyme substrate recognition. In addition, aspartate-arginine salt bridges are involved in the stabilization of protein structures and the modulation of enzymatic activity. Understanding this interaction is essential for drug design, protein engineering, and understanding the mechanisms of various biological phenomena. Aspartate kinases are enzymes that catalyze the phosphorylation of aspartate, yielding beta-aspartyl phosphate. This reaction plays a key role in the regulation of aspartate metabolism and is a crucial step in the biosynthesis of certain amino acids, such as lysine, threonine, and methionine. Dysfunction of aspartate kinases can lead to imbalances in amino acid metabolism and interfere with essential biological processes. Aspartate transporters are membrane proteins that facilitate the transport of aspartate across cellular membranes. These transporters are vital for maintaining the intracellular aspartate concentration, ensuring proper cellular function. Various families of transporters, such as the aspartate/glutamate transporter family, play important roles in neurotransmission, amino acid metabolism, and gut-brain communication. Asparagine biosynthesis is a metabolic pathway that converts aspartate into asparagine. This pathway involves the action of enzymes such as asparagine synthetase and aspartate ammonia lyase. Asparagine is a non-essential amino acid that plays critical roles in protein synthesis, nitrogen storage, and cellular signaling. Dysregulation of asparagine biosynthesis has been implicated in certain cancers and metabolic disorders. Aspartate plays a significant role in the regulation of the urea cycle, a series of biochemical reactions that occur in the liver, aimed at detoxifying ammonia, a byproduct of protein catabolism. Aspartate is involved in the synthesis of citrulline, a key intermediate in the urea cycle. This process ensures the efficient elimination of toxic ammonia from the body and maintains nitrogen balance. Aspartate oxidase is an enzyme that catalyzes the conversion of aspartate into iminoaspartate, which subsequently undergoes non-enzymatic hydrolysis to form oxaloacetate. This reaction generates hydrogen peroxide as a byproduct and has been explored for various biomedical applications, including cancer treatment and biosensors for hydrogen peroxide detection. Metabolites derived from aspartate metabolism can serve as important biomarkers in various diseases. For example, elevated levels of aspartate aminotransferase and aspartate-derived metabolic intermediates, such as oxaloacetate and alpha-ketoglutarate, can be indicative of liver dysfunction, heart disease, and certain cancers. The measurement of these biomarkers aids in disease diagnosis, prognosis, and monitoring treatment response. In conclusion, aspartate, a fundamental amino acid, plays multifaceted roles in biochemistry and medicine. From its involvement in various metabolic pathways to its clinical significance as a diagnostic marker and therapeutic target, the study of aspartate unravels the intricate web of biochemical processes underlying normal physiology and disease states. By understanding the complex mechanisms and functions of aspartate, we gain valuable insights into the complexities of human biology and pave the way for novel therapeutic strategies.